Hello my dear students, today we will discuss C4 pathway in plants. As we have seen in our previous class that the first carbon dioxide acceptor in C3 cycle is 3-PGA, 3-phosphoglyceric acid. Now what are the C4 plants? Which is the first carbon dioxide acceptor in C4 plants, which is the first stable product formed in C4 plants. Today we will discuss all in our class. So my dear, C4 plants are present or found in tropical areas. Means they prefer to live in the high light intensity areas or high temperature areas. So this pathway is also known as Hatch and Slack pathway. Hatch and Slack pathway, because this was C4 cycle was studied by two biologists, Hatch and Slack, and that is how the C4 plants are also known as Hatch and Slack pathway. This pathway was discovered in sugarcane. Now my dear students, the first carbon dioxide acceptor in C4 pathway is phosphoenol pyruvate. This phosphoenol pyruvate, or in short you will write down it is PEP, PEP. It is phosphoenol pyruvate, a 3 carbon molecule. 3 carbon molecule. So my dear students, as earlier I told you, in the C cycle the carbon dioxide acceptor is 3-PGA. Now here the first carbon dioxide acceptor in C4 pathway is phosphoenol pyruvate. It's a 3 carbon molecule. And the first stable molecule or compound, the first product which is formed is 4 carbon molecule that is known as oxaloacetic acid, OAA, oxaloacetic acid. Now my dear students, as we have seen in the anatomy that there are dorsoventral leaf and there are isobilateral leaf. Isobilateral leaf means monocot leaf, and dorsiventral leaf means that's a dicot leaf. Now in the dorsiventral leaf, stomata are majorly present in the abaxial side, means in the lower side of the epidermis. But in case of the isobilateral leaf, that is a monocot leaf, stomata are present or they are distributed equally. Means where they are found on the upper side of the epidermis, they are found on the lower side of the epidermis, that is abaxial, and upper one is addaxial side. Now my dear students, if I will make a cross section of a leaf, as you know, this is the addaxial side or the upper side, that is the upper epidermis, And here is the, we can say, the abaxial side where stomata are majorly found. So I will show you a stomata here. Now in case of dorsiventral leaf or in case of dicot leaf, my dear, mesophyll is differentiated. Mesophyll or mesophyll cells, a tissue, are differentiated into palisade parenchyma and spongy parenchyma, right? And spongy parenchyma. So this is in the dorsiventral leaf or in the dicot leaf. But the case is different in the isobilateral leaf, or we can say in the monocot leaf the case is different. Here in the isobilateral leaf, mesophyll is not differentiated. So we can say that mesophyll is undifferentiated into palisade, differentiated into palisade and spongy parenchyma. So we will say that simply that in isobilateral leaf, mesophyll is not differentiated or mesophyll is undifferentiated. Now this is a case with the C3 plants. Now in the C4 plants, what happens? So these are the vascular bundles, means xylem and phloem. Collectively we call them vascular bundles. Around the vascular bundles you will see a large sized cells. They are known as bundle sheath cells. Right? So I will write down here, these are bundle sheath cells. And in case of the C4 plants, there are mesophyll cells. You will find they are loosely arranged. So these are mesophyll cells. So this is the upper epidermis, this one is the lower epidermis, right? This is upper epidermis, this one is lower epidermis. And you can see here the mesophyll cells. These are bundle sheath cells. That is why, my dear students, in C4 plants, there is a special anatomy that is found in the C4 plants, like in the sugarcane, maize, sorghum, atriplex, that there is Kranz anatomy. Remember this word, that is Kranz anatomy. So this Kranz anatomy, this you will not found in the, or this is not present in the C3 plants. In the C3 plants, there are mesophyll cells. Mesophyll cells, they contain chloroplast. And hence in C3 plants, we can say the photosynthesis, or we can say the carbon dioxide that is reduced to the glucose is taken place in the mesophyll cell. So my dear doctors, but the case is different here in the C4 pathway. Here you will see a ring-like structure around the vascular bundles. These are the bundle sheath cells. This Kranz means, it means wreath or wreath. So mesophyll cells, these mesophyll cells are loosely arranged. They are loosely arranged. However, in bundle cells they are compactly arranged. My dear, they are compactly arranged. Now if I will make a difference here, or we will differentiate this mesophyll and bundle sheath cell here, that these are bundle sheath cells, And here I will write down the mesophyll cells. I will write down the mesophyll cells, right? So these are bundle sheath cells. I will make a difference here, bundle sheath cells, and then we will make a difference between this bundle sheath and mesophyll cells. Mesophyll cells. Now, my dear, as you have seen in case of a transverse section of a leaf, where you have seen that mesophyll cells are loosely arranged while the bundle sheath cells are compactly arranged. So the same thing we will write here, that we will write down here that bundle sheath cells are compactly arranged. They are compactly arranged, my dear. They are compactly arranged, right? So in case of mesophyll cells, we will write down that mesophyll cells are loosely arranged. They are loosely arranged. It means that intercellular spaces are present. Intercellular spaces are present. So here in the bundle sheath cell, my dear, we can write that intercellular ICS are absent. Right? Let us do it the third point, that the Kranz anatomy, which I told you earlier, that is a special feature of a C4 plant. And here in the bundle sheath cells, as I mentioned, they are compactly arranged, intercellular spaces are absent here. But my dear, you will see the world's most abundant protein, that is rubisco. That is rubisco is present. That is present here in the bundle sheath cells. The world's most abundant protein, that is rubisco. That is ribulose, bisphosphate, carboxylase, oxygenase. It has a dual nature. During the much more concentration of carbon dioxide, rubisco combined with the carbon dioxide. Now when there's more oxygen, rubisco shows the affinity with the oxygen. So my dear, as you know, the C3 cycle that happens or that occurs in the stroma, while as the light reaction takes place in the thylakoid or in the grana. So in the C3 cycle, that occurs in the stroma of a chloroplast. Because the products which are formed during the light reaction, like ATP, NADPH, they are utilized here in the stroma during the dark reaction. Now, my dear, so I will write down here, rubisco is present, but there is one enzyme, that is pepcase, that is phosphoenol pyruvate carboxylase is absent here. So it means that food synthesis cannot take place here in the bundle sheath cells. No doubt they have chloroplast. In the C3 plants, mesophyll cells are responsible for the synthesis of food. But here in the C4 pathway, you will see how the transformation of the compounds takes place from mesophyll cell to the bundle sheath cells. So here, cells are compactly arranged, intercellular spaces are absent, rubisco is present, pepcase is absent, and both pigment system first and pigment system second, that means PS1 and PS2 second are present. In other words, it simply means that they show the non-photophosphorylation. because both the pigment system 1 and pigment system 2, they are involved here. But my dear, in case of this, only PS1 pigment system 1 is present, right? And here I have written ribosco. Ribosco is absent here. I will make it show with the cross. But the PEP case, that is phosphoenol pyruvate carboxylase, is present. In other words, my dear, it simply means that photosynthesis takes place. Photosynthesis takes place. So I have written here ribosco is, I am writing about the bundle sheath cell. So I have written here in the bundle sheath cell that ribosco is present, PEP case is absent. So I have written here ribosco is absent, but PEP case is present. Okay, so let us do it diagrammatically now. So I will show you with the diagram how this, transformation of the compounds takes place and how food is synthesized and carbon dioxide is reduced. So let us make a diagram, my dear. So you can draw a diagram with me. So these are two different cells. These are two different cells. One is mesophyll cell and second one is bundle sheath cell. Okay, so you can draw with me. Now, so these are now mesophyll cells. Mesophyll cells. So these are bundle sheath cells. Bundle sheath cells, right? So now you can see now the atmospheric carbon dioxide, right? Because photosynthesis take place in a leaf. There are mesophyll cells in a leaf, and in the mesophyll cell you will see the chloroplasts are there. There are grana, there are stroma, and in the grana or thylakoid, light reaction of photosynthesis takes place, and here in the stroma, the dark reaction of photosynthesis takes place. It never means that photosynthesis takes place during night. No. So this is a misnomer that is used here with the dark. Actually, the products which are formed during day, because light is sufficient available, ATP is synthesized, NADPH2 is synthesized, and oxygen is released there that goes into the atmosphere through the stomata. And you can see now this atmospheric carbon dioxide, once it enters into the mesophyll cell, as I mentioned here, mesophyll cell. So there in the mesophyll you will see water molecule. With the addition of the water molecule, this will form HCO3 carbonate, right? And my dear, in the mesophyll cells, as I have written in the mesophyll cells, that PEP carboxylase is present. So the first carbon dioxide acceptor in the mesophyll cells or in the C4 pathway, that is phosphoenol pyruvate, in presence of an enzyme, that is peptase, that is phosphoenol pyruvate carboxylase. So dear students, my dear students, atmospheric carbon dioxide goes inside through the stomata. It gets a molecule of water plus the first acceptor of the carbon dioxide, that is PEP, phosphoenol pyruvate, in presence of enzyme, that is peptase, and this will form the first, that is the 4 carbon product, the first stable product, that is oxaloacetic acid. Now this oxaloacetic acid will get reduced and this will form or this will change into a malic acid, once again a 4 carbon compound. So as we have seen that the bundle sheath cells are impervious to gases. So gases cannot enter directly through the bundle sheath cell because they are impervious, my dear. But the mesophyll cells are pervious to gases. That means carbon dioxide can enter, oxygen can leave, but this will not happen in the bundle sheath cells. That is how the carbon dioxide is trapped with the addition of the water molecule. This will form the oxaloacetic acid in the presence of an enzyme that is peptase. And my dear, this oxaloacetic acid will terminate into the oxaloacetic acid. Then with the reduction, this will terminate into the malic acid. Now, or the aspartic acid. Now, this malic acid will enter into the bundle sheath cell. So this malic acid will, the 4-carbon compound will enter into the bundle sheath cell now. So that means the transformation and this malic acid enters into the we can say bundle sheath cells, transformation. So one, you know my dear, that malic acid is once again a 4-carbon, right? So what will happen here? Decarboxylation will take place. Decarboxylation. Decarboxylation means release of the carbon dioxide. So instead of receiving the direct carbon dioxide, that is the characteristic feature of the mesophyll cells. But the bundle sheath cell is inside the bundle sheath cells, carbon dioxide is also produced. So this is the 4-carbon. With the help of decarboxylation, 1 carbon dioxide gone, and what will happen now? There will remain only now the 3-carbon, and this will form a product that is known as pyruvate or pyruvic acid. That's a 3-carbon, as you know, CH3 COOH. This is the formula of the pyruvic acid. That means there are 3 carbon, 1, 2 and 3, there are 4 hydrogen and there are 3 oxygen. So this is a 3-carbon now, this pyruvic acid, my dear students. And now this pyruvic acid, this will once again transport into the mesophyll cells. Okay, and in the presence of an enzyme that is pepsynthase, so this 3-carbon, that is pyruvic acid, will terminate into once again convert into the phosphoenol pyruvate in presence of this pepsynthase. So it needs here some phosphates, from where this will get the energy. It will get the energy from 2 ATP molecules, and this will form ADP, and this will get phosphate and will form a PEP, that is phosphoenol pyruvate. My dear students, my dearest students, what will happen with the carbon dioxide now? So you see the carbon dioxide in the stroma. As you know, I have written there in the bundle sheath cells, ribosco is present. That is ribulose, bisphosphate, carboxylase, oxygenase. And this will form what? This will form once again. So this will form in presence of an enzyme ribosco that is found in the stroma of a chloroplast, and in presence of RUBP and this ribosco enzyme, what will happen here? So once again a C3 cycle is occurring. C3 cycle is happening. So if somebody will tell you, hey, is C3 cycle happening in the C4 pathway? Yes. You will see in the bundle sheath cells, Calvin cycle once again will occur, once will happen, and the photosynthesis will take place. But photosynthesis is not taking place in the mesophyll cell in the case of the C4 pathway or in the C4 plants. So my dearest, so you can, as in case of the C3 cycle, what we have deduced the result that what we have got the result that for one carbon dioxide fixation, okay, there we need 2 NADPH2, right, and 3 ATP, but the case is different here. So here in the C4 pathway, for one carbon dioxide fixation, okay, so it will be 1, 2, and 5. You can multiply with them 6 because the cycle has to go 6 times, because this is a 1 carbon, and here in the glucose you will see there are 6 carbons. So what will be the overall result? Then you have to multiply with the 6. That means 6 ones are 6, that is 6 carbon dioxide. We require 6 to 12, we need 12 NADPH2, then 6 5s are 30, 6 5s 30, then it is 30 ATP. And out of 30 ATP, you will see that 18 ATP are already produced there in the C3 cycle. So I hope you got the complete idea and you have grasped this C4 cycle completely. Thank you very much for listening patiently. Best of luck. Thank you, my dear students. Allah Hafiz.